Hey Year 10s! So here's our practice test for cells. A couple of questions we didn't learn about and they're not going to be on the test, so I'll let you know which ones those are. But here we go, let's get into it. So the first few questions are multiple choice. Firstly, cells are the basic building blocks of, well, they're the building blocks of every living thing, so it's definitely C in this one. Both plants and animals. When you alter any of the adjustment knobs on a microscope, so the coarse focus or the fine focus, you're definitely looking through the eyepiece lens. So when light travels through the two lenses, remember there's the eyepiece at the top, and that's times 10, and then you've got objective lenses at the bottom, and you're picking one of those three. And in this case, the objective lens we're using is times 20. Remember, we multiply the numbers together to get the total magnification. So that is 200 times in this case. The next question, the field of view gets smaller when the magnification decreases, increases, or remains the same. I don't think I mentioned field of view, but it's the total view of what you're seeing under the microscope. So our field of view is normally round, and you're seeing whatever cells are in there. But if we zoom in to that, then in the next case, after we've zoomed in, then we're only going to be able to see less or fewer cells than we saw originally because we've zoomed in. So the cell, what we see will get bigger, which means the total field of view, in other words, the distance across that we're looking at, gets smaller. So field of view increases as the magnification increases. So when you zoom in, you can see less. Alright, a drop of pond water is going to be viewed under a microscope. Firstly, once you've collected your drop of pond water, you need to put it on a slide. That's the glass, piece of glass that we use to put our samples on. A cover slip goes on top of that. We didn't actually get a chance to use cover slips, but they're the piece of plastic or glass that goes on top. On the control center of a cell, we should remember that one. That's definitely the nucleus. Prototyster, or the other name for this one is Prototyster. It includes all the protists, and they're a range of different species. They can either be single-celled or multi-celled, but they're more complicated than bacteria. So their cells have lots of organelles in them, whereas bacteria are really simple. So that's a bacteria. A protist cell is normally much larger. It's got a nucleus. It's got mitochondria. It's got vesicles. Whereas bacteria only have ribosomes. Really simple cells. So Prototyster is a group of organisms made of single cells. Possibly. It doesn't contain animals. It doesn't contain bacteria. It pretty much is species that don't fit into other groups. You could argue it's made of single cells, but not all protists are single cell. Okay, next. The green pigment in plant cells. Pigment is just a colored chemical. And this is the pigment that's found in the chloroplasts of plant cells, and it's called chlorophyll. The next one is, which of the following is the biggest difference, or is one of the biggest differences between plant and animal cells? Plant cells have a cell wall and animal cells don't. Definitely the first one. The position of the nucleus, no, that can change in all different types of cells. every cell has cytoplasm, so it's not that one, and every cell has a cell membrane, so not D either. Alright, parts of the microscope. We've used them, we should know all these. A is the eyepiece, lens. B here is the coarse focus. C is the stage where we put the slides on. D is the base. Even though this is a really old microscope, you can still tell which part is which. Although for E, you can see this microscope's so old it actually has a mirror. So you need to have a light source facing sideways, so some sort of lamp, and it needs to send a beam of light to the mirror here, and then the mirror reflects the light up through the microscope. So normally E is the light source, in this case it's the mirror. Light, you can also call it a lamp if it has a globe. Okay, next one. 2 tips for effective use of a microscope. Well, we've talked about lots of different ones when we use them. One would be turning the head so the eyepiece is facing the other way. So if we have the base, we have the frame going up to the head. We want to turn the head around so the eyepiece is facing backwards compared to how we store them. That would be one tip. And there's the stage and coarse and fine focus. So we could use the eyepiece facing the other way. Facing backwards. What else? We should remember that we don't turn the coarse focus once we're beyond the first objective lens, because that can move the stage up too much and then crack the slide. Don't use the coarse focus beyond the ×4 objective lens. So remember, there's ×4, there's ×10, and there's ×40. Okay, that would be 2 we could do. Plenty of other ones. But we'll stick with those 2 for now. Okay, draw and label a simple sketch of a human cheek cell. Okay, so I'm looking at this. A human is an animal, obviously. So we're not going to have a cell wall. So let's follow all our sketching techniques that we learnt about. So I'll draw a simple cell membrane. And already I've done a bit of a sketch. Simple cell membrane. It needs a nucleus and it needs cytoplasm. Well, nucleus is somewhere in there, and we're using stippling to shade it in. And it will have the cytoplasm, and we can shade it or just leave it actually. Okay, and then we're using straight ruled lines, horizontal, to show all of our features. Nucleus, cytoplasm, if I could fit it in, and cell membrane. They didn't actually tell us in this question whether which magnification we've used, but we'll give it a title anyway. So we'll say human cheek cell for our heading. That's all we need. And we could say magnification unknown. All right. Question 13. Name 2 of the 5 kingdoms that every living thing might be grouped into. Well, hopefully you know all 5. We've got Kingdom Prototyster, Kingdom Monera, which are the bacteria. We've got Animalia, which are the animals. We've got Plantae, which are the plants. And Fungi, which everyone always forgets, which are the mushrooms and all the other fungi. Any 2 of those. Okay, what are mitochondria and what are their purpose? Well, mitochondria are one of the organelles. So that would be 1 mark here. Their purpose, well, the common saying everyone remembers is they're the powerhouse of the cell. But what they're really doing is they're converting all the food energy, all the food molecules that we eat and that our cells absorb, converting that food into energy for the cell to use. All right. Next question is, why are there different types of animal cells, i.e. differentiated cells? Differentiated is the same thing as specialized cells. So why are so many cells specialized in animals? Well, the whole reason that cells specialize is because they all have a certain job to do. So there's 4 marks for this one. So I'd probably say that, firstly, animals are multicellular. And because they're multicellular, so therefore, our bodies are organized, as we know, into not just a whole bunch of cells, but they're organized into tissues, and then into organs, and then into systems. So therefore, the cells are organized into tissues, organs, and systems. So then, that means that cells need to be specialized so that they can perform the job of the organ or the tissue that they're in. Cells need to be specialized so they perform their role. Or in other words, their tissue's role, whatever it's meant to be. So a muscle cell needs to be really good at, a muscle cell's specialized so that it can move, which most cells can't do. Okay, and the last one is probably that there's lots of different types of tissue, so therefore there has to be a lot of different types of cells. There are many organ and tissue types. So therefore there will be different types of cells. Many different specialized cells. So 4 marks, we've made 4 points of information and we've summarized it in the bottom, and we've used therefore to show how our logic links together with our ideas. Many different specialized cells. Okay, take a screenshot of that if you need it, otherwise it's gone. Okay, the next one, copy and complete the sentence. We've got the large cavity in the middle of a plant cell. Well, we should remember that. Plant cells have a single large vesicle, which is called a vacuole. And it contains a fluid, which we didn't learn the name of and you don't need to know. It's called cell sap. I actually had to just look it up. It's not a commonly used phrase. Okay, number 17. Label these common cell structures. And cytoplasm is the same as cytosol at our level. So we want to label... So we want to label the nucleus, the cytoplasm, or cytosol, and the cell membrane. Okay, so we've got Paramecium and Euglena here. 2 species of single-celled organisms. So the nucleus generally is the dark organelle in the center or towards the center. Let's get some arrows working properly. There we go. Nucleus, and in Euglena, there's our nucleus. The cytoplasm is just anywhere in the open space of the cell on the inside. And Euglena, it's a bit difficult. Okay, and then cell membrane. Well, obviously that's on the outside. Shouldn't be too hard. 1 and 2. Okay, so these cells are single-celled, but they've got complex organelles inside, like a nucleus, and we can see some mitochondria. Actually, these are chloroplasts over here in Euglena. So if they're single-celled but they're complex, then they have to be protists. So straight away we're thinking, okay, these are protists. At level 10, we learn how to compare the difference between eukaryotes and prokaryotes. Prokaryotes are only the bacteria. Every other kingdom are eukaryotes. So if these cells are protists, then they have to be eukaryotic, or they have to be in the group called eukaryotes. Okay, question 18. Complete the table below to show which organelles you'd find in each cell. Okay, nucleus. Both animals and plants have a nucleus. Cell wall, only plants do. Cell membrane, well, every single cell has a membrane, otherwise the cell would fall apart. Same with cytoplasm, every cell has liquid in it. Large vacuole, we should know plant cells have one, animals don't. And the same with chloroplasts. Alright. Give 2 ways in which prokaryotes are different from eukaryotes. So basically we're comparing how the bacteria, which are the only prokaryotes, how are they different to all the other kingdoms. And I've mentioned a few. You learn a lot about it at level 10. So what do we have? Prokaryotes. This question just says give, so we can list them. That will be enough. Oops. So prokaryotes have no complex organelles. And no nucleus. And eukaryotes are the opposite. So they do have complex organelles and they do have a nucleus. Complex organelles, I mean things like mitochondria, vesicles, endoplasmic reticulum, and Golgi apparatus. Prokaryotes have none of those. The only organelle they do have are ribosomes, which are one of the very first organelles to evolve. And that's why every single living thing has them. So they do have ribosomes. Okay, describe 3 of the differences between plant and animal cells. That's basically repeating the question from above. So if you had have mentioned plant cells have a cell wall and they have a large vacuole and they have chloroplasts, that would answer that question. Okay, the last 2 are to do with viruses, which we didn't focus on, and it's not on the test. For your extra information though, are viruses alive? Well, it's pretty much debated. I like to say no. I like to go by the definition of life to mean made of cells. So, in other words, you can call life cellular life. Back in year 7, remember we learnt an acronym. I think it's Mr. NC GRU. Well, there's lots of other ones. GRU. So in that one, the C stands for cells. This is the acronym for how do you know if something's alive or not. There's move, respond, takes in nutrients, grows, and so on. If we're going by the cellular life definition, viruses are not alive. So I'd say no, because of the reasons I just said. Because they're not made of cells, so they don't fit in with the whole other group of life, which is made of cells. Explain the structure of a virus. You definitely don't need to know this, but for your interest, viruses have some sort of outer shell, which is made of proteins. Many viruses are this sort of 3D hexagonal shape. I think it's called an icosahedron in 3D. So there's a coat of proteins around the outside. Again, it's not a cell membrane because they're not made of cells. And then on the inside, they just have some genetic material, which is either DNA or RNA, and we'll learn about the difference between those later in the term. And that's all that viruses have. There is some more detail, like their proteins do stick out. There are a few different types of proteins in the protein coat, which help them to attach to their host cell. But other than that, they're just made of protein plus DNA or RNA. But we don't need to know that for the test. Alright. So check your answers, use that for revision, and I'll see you on Tuesday.